course, I have to ask, and you can give me a thumbs up or something if you can see the screen share. Excellent. Good to know. We're going to give it just a few more seconds here. We'll give them a full minute. I think the only thing that's weird about these Zoom talks is I'm very much into like a discussion kind of situation, and I just can't get the audience participation aspect right during Zoom meetings, because there's like too many people talking at one point, or it's kind of interesting. I would say it's my least favorite part about this is that we can't just like see each other and actually like cheers.
I'm actually gonna go get a new beer because mine has, I think it's pretty diacetyl-y.
No.
I'm like sitting here trying to like decide if it is or isn't, because sometimes I feel like I fake that I taste it, but I'll be right back.
Does it feel really like slick and smooth?
It doesn't feel like the most slick and smooth, but it does a little bit, and it's like buttered green pepper kind of. Yeah, and it's like more and more, the more I drink it, the more I taste it. So
yeah, and I'm sure the more it warms up, the more you taste it.
That's true too.
Right. I think as soon as Dre gets back with a fresh beer in hand, we'll go ahead and get started. First of all, feel free to throw any questions in chat. I'll try to answer. I am doing Q&A session. This presentation only lasts for about half an hour or so. And I will be doing Q&A. So feel free to ask questions right away or save it for end of the situation. I am going to go ahead and get started. Okay, so hopefully by now my name is Dana Garvis. I run Oregon BrewLab, which is a very small beer testing facility inside Eugene, Oregon. And today I'm going to talk to you about using the microwave to sterilize your micromedia in the beer lab. And just as a quick overview for Fireside Chats, I hope to have once a month some sort of little half-hour presentation similar to today about just a topic that is interesting to me and hopefully maybe interesting to you too as well. So let's get started. So just a quick overview of what we're going to be looking at. We'll look into what spawned this, sort of what science has to say about microwaving media, and then looking at different comparisons to the autoclave and the pressure cooker with microwaving in mind. And then looking at my own experiment that I ran in order to sort of test this theory. And then we'll talk about the conclusions we draw from that and further exploration. So what started this for me really is this summer, because of COVID, I was able to spend way more time at home, which meant that my garden was bomb. I had so much tomatoes. I had tons of habaneros this year. They did really well. But one of the things that really grew well this year with my extra attention was my expansive herb garden. And so did my friends' gardens. And so right around Mother's Day, I had this idea that I wanted to make like an herb de Provence mixture to send my mom for Mother's Day. And I wanted it to be from my garden and my friend's garden. So I was looking for quick, dry herbs on Google. How can I do this as fast as possible? Because I have a whole bunch and I don't really have time to sit around and wait for them to dry traditionally. So I Googled it, and America's Test Kitchen, something I think we all can agree is somewhat reliable, said you should use the microwave. And so I did. I used the microwave. You just take 2 pieces of paper towel, put the herbs between it, spread them out a little bit. You can destem the herbs or not. In 30 seconds, and you have dried herbs, and it's fantastic. And so this kind of got my brain working about, well, why does this work? You know, you've probably seen this graphic in some sort of science class you've taken. Visible light is an area we can see kind of in this area. If we go higher than that, we get into an area that can cause damage to our bodies. But along the electromagnetic spectrum, the longer we let these wavelengths get, the less harmful they become, right? So microwaves kind of sit right here in the middle. And the way that they work, I'm not going to get into it too much, but basically they vibrate water molecules enough to the point where they can start boiling and even evaporate. And so I did this with herbs, and I started thinking, well, wow, that's a really incredible tool. Why did I not think about something like this before? I wonder if I could do this with media broth. Like, I wonder if this is something that I could take into the lab. It's not going to be too expensive because microwaves are a dime a dozen. You can find them for 10, 20 bucks at a secondhand store, or you can get them on Amazon. So I started actually looking into, well, what do the alternatives cost? Well, an autoclave is anywhere from $800 used on eBay to $3,000 for like one of the smallest models possible, brand new. So you're looking at huge, huge price cost to run an autoclave. Not to mention, your running time takes about 60 minutes between getting everything all up to pressure and temperature. Then you want your 15-minute time at that temperature and pressure. Then it takes about an hour to 2 hours for that to cool down and actually depressurize so that you can reach in to grab the media. And, you know, capacity varies. On the small end, you can only fit 1 or 2 flasks. On the large end, you can fit a few more larger flasks. It's relatively safe considering the extreme pressure and heat. It does have shutdown features, which is one of the like really beneficial parts to an autoclave. Pressure cookers are a little more expensive or a little more cheap. You can get into some very expensive pressure cookers, but you could probably find one for about $200 looking online. They take, again, about an hour, a little less if you're really proficient with it, to get up to pressure and temperature. The cool down time is a little quicker than the autoclave. And I think between the budget and the run time, not to mention its massive capacity, as you can see there, I think that that's part of the reason why the pressure cooker has become sort of a staple in the brewing lab right now. One of the things I do not like about the pressure cooker that I think most technicians who run micro using a pressure cooker will tell you they don't like is that there's extreme heat and extreme pressure and you cannot walk away from it. As soon as you purchase one, you are on lists for the FBI and Homeland Security. It has not impacted my life that much, but it is a consideration. Whereas here we are at a microwave. I had to work hard to find a $200 microwave, and this is a microwave that is way beyond anything you would need in the lab. You know, it's voice activated. It has multiple convection waves. Like, it's top of the line. So you could get that one, or you could spend 60 bucks on the Amazon basic microwave that everyone has. It's really perfect, and it's what I purchased in order to run this experiment. And it turns out the yeast that I bought was more expensive than the microwave. In total. So what's cool about the microwave, it only takes 8 minutes, maybe 10 if you have a heavy load of media in there. You know, the capacity isn't quite the same as a pressure cooker, but because of how quick it is, you can do multiple batches in the same amount of time. There is no shutdown feature, so I suppose that could be considered a safety issue with the microwave. However, it's also not a bomb threat. So I really like the microwave, and I'm really starting to get into this idea. So I looked into precedents. Where in microbiology history do we actually see people looking into microwave usage? And it turns out that we've actually been doing it since about the 60s. This is the model of the microwave that was used in this paper that I have referenced here from Latmir Matson. This is a model that's like a year or two older than the one they actually use, but it's the only one I could find a cool little ad from. I think it's pretty sweet. So what they looked at is they actually looked at using microwaves to decontaminate samples. And they looked across a bunch of different bacteria types. And they were the first to like really look at just sort of like throwing it in the microwave for a few minutes. But before that, we had Goldblythe and Wang here, who started looking at smaller cycling time. So they'd only put them in for a minute, but they did that 6 times. And they found that they were able to completely kill E. coli and this other bacteria in pieces of gauze that were inoculated. So then we get into some really weird shit in 1969, where one study from Lichowich and Beerchat actually literally wanted to see, is it the heat of the microwave or is it the microwaves themselves that is causing things to actually sterilize? And so the way that they did this was they put a condenser system inside the microwave and suspended kerosene at -25°C so that they could counteract the heat of the microwave, just to prove, yes, just microwaves can kill bacteria and sterilize contaminated media. But it also is the first study to introduce our favorite brewing yeast here, Saccharomyces cerevisiae. So now we're seeing microwaves can kill yeast. Awesome. Kulkin and Fung found a way better way to do this without having to use kerosene at -25°C. And they were able to prove, again, that decontamination went through for E. coli and Salmonella with just microwaves. So we're pretty well established that the heat and the microwaves are things that can both sterilize. And then here, Vila and Wu in 79, they found that heating your samples in water and without water still both will decontaminize your media. So this is all really exciting because I think that it can pertain to beer. Let's jump ahead a little bit. This is Logan and Dvorak in 1999. They did a study on just the history of using microwaves in microbiology. And this was a huge like jumping point for me. I was able to go back quite far and get an idea, but I think ultimately moving forward is where we want to go. So what we found is that Border and Spearman in 99 were the first to actually do a direct comparison between autoclave and microwaved medias for bacteria and yeast. And I thought that their like results was just so good. I had to put the whole thing in here, even though I don't expect you to read it. It basically says yeast cannot survive in a microwave for longer than 15 seconds. And what's really important about this study in particular and what pertains to beer is that it says here, after 3 minutes in the microwave, powdered prepared media was free of contamination while also able to support growth. That's huge. What that means is that you can sterilize your media in a microwave and like actively get rid of any potential inoculations. And then later you can use that same media to grow up any bacteria or yeast you want. That's amazing, right? So we jump ahead a few years and now we're starting to look at the biodiversity of using microwaves. Does microwaved media look different than autoclaved media? And Kothraki and Petita in 2011 showed that microwave sterilized media actually supports better microbial growth than autoclaved media. So basically, not only is the microwave cheaper and faster, but it works better. That's really exciting. And then, you know, the most recent thing I could find about microwave media is actually from Schlepper in 2014, who writes a procedure for non-clinical labs using sterilized media. This means that it's been completely adopted down the line from, you know, grade school. She has a little procedure for people who are in 6th grade to like look at the bacteria from their saliva, you know, up to high school, and then also in undergrad labs. And we're also seeing now in labs where you don't have a human perspective part of it. So, you know, you can use a microwave for anything except for, say, like dental. You don't want your dentist using a microwave to sterilize your stuff. And of course, you cannot sterilize metal in a microwave. All right, so what does that mean for us? Well, that means that we know microwave media is a good way to go about getting our culture plates ready. What we don't know for sure is how well brewing bacteria and brewing microorganisms hold up to this regimen. So my hypothesis is, does microwaving media effectively sterilize and grow brewing microorganisms as well as pressure cooking media? And so this is how I did it. I'm sure you can't tell any of these yeast suppliers at all because I blocked out their names. But my local homebrew store gave me a whole bunch of extra stuff. So I got 8 or 9 different strains, and they gave me a few from their graveyard because they were so interested in seeing how this would progress. So I went into it with 8, came out with 11. I'm looking at pressure cooker because I do not have an autoclave. They are so expensive. And so a pressure cooker is what I have used for just about 10 years in the brewing industry. And I'm also going to look at microwave media. Then what I did is I used WLD, which is Wallerstein differential media. I used UBA, which is universal beer agar. And then lastly, I used HLP, which is Sue's Lactopedia selective. Now the microwave can only hold 2 of these larger jars, so the HLP was done in a different time. Then what I did is I plated, photographed, and gram stained all 110 samples of each of these 11 yeasts, Brett's, Lactosepedias. This is one round of gram staining, which helps me identify and take really nice bacterial photos. All right, so how does this work? I'm not going to go into how you plate media. There's tons of talks out there you can find on YouTube, or more beer specifically, you can find at the ASBC website, which is the American Society of Brewing Chemists. I highly recommend these 3 SOPs. Microbiological control talks about how to take samples aseptically and also how to just do plating. Takes you through the actual steps. Microbiological control number 4 talks about using different general medias such as UVA. And then microbiological control 5 talks about differential medias such as WLD and HLP, which I used. So in general, how we're going to do this: place 1 to 2 bottles into the microwave. Make sure the lids are fairly loosened before putting in there. And when you're making your media, make sure you have at least 50% headspace. These things can get really foamy, and especially with the universal beer agar, that does require a little bit of beer in the liquid media. If that's not degassed, that can actually get really foamy really fast. So this is just a quick little video of what that looks like. Yay, it's super foamy and bubbly. So on the microwave, set your timer to 5 minutes. You're gonna let it go until you have your first sign of boil. Then you're gonna open the door, reset the timer to 3 minutes, and you're gonna let that finish out, stopping every time the boil comes close to the edge of the head space there. It takes just about 8 minutes. Here is a time lapse that I'm gonna talk you through just a little bit, even though a lot of it's waiting. There's no way you could do time lapse, I think, of pressure cooking because it is such a long process. So this whole process, you can maybe see in the corner here before I started, It was at 10:56, and I finished this whole process. You'll see that the clock reads 11:05. So it took 8 minutes in real time to make enough media for, I think, let's see, 80 sleeves of plates, or sorry, 80 plates, 4 sleeves. Then, 5 minutes on the timer, and it takes about 2 minutes to hit the boiling factor. Then after that, you hit your sterilization time period, which is exactly 3 minutes. And that boils us down to a 1-minute video. So like I said, there's no way you could do a 1-minute video with how long it takes for media to cook in a pressure cooker. So then you're gonna wanna pour your plates aseptically. I like to take lovely photos. I think these are particularly artistic. And then I like to label them. I use this particular brand, clear labels, so that I can take photos of the plates. Again, I'm not gonna go into how you plate, but this is like a nice little graphic that kind of talks about the difference between pour plates and streak plates. And however you decide to inoculate your plates, do it that way, aseptically. You're gonna wrap those in parafilm strips, and then we're gonna incubate. Whether or not you choose to have an incubator is up to you. You can leave it in a room that tends to stay nice and warm in its own sealed container. You can see here in my incubator even, I have a anaerobic jar, which houses basically a fancy hand warmer. And those hand warmers, the way they work are they have a chemical in there that absorbs oxygen and releases CO2. So when those are encased in a sealed jar, it creates an environment that is anaerobic, which means without oxygen. And then here you can see, I just have another little box for aerobic, meaning with oxygen. And this is, a little $400 unit incubator. Not too expensive. I did get the analog, so it'd be cheaper, which, that's your own preference, I think. All right, so let's look at some of this data. It's a little bit overwhelming because there are 110 plates, and I take a photo of every single one, except for the anaerobic ones, every day for 7 days. So these are screenshots of my Google Docs page. And obviously day one, this is only maybe half the data. So you can see the WLD is this nice blue media. You can see the UVA is this really nice tan, kind of orangey media. And you can see these interesting plates here that kind of have both colors. And that's because WLD, again, is a differential media. So bacteria that grows that produces acid, like lactic acid bacteria, will actually stain these blue plates a different color. And it basically is just another check that says, yep, I know that this is lactobacillus, or I know that this is pediococcus, both of which are lactic acid forming bacteria. So look at, there's 50% blue, 50% orange here. Day two, yeah, this data is getting pretty redundant, but you can see some of these blue plates are actually really starting to lose their entire color. Day three, those pictures aren't as good for some reason. Let's skip ahead to day 5. Wow, you can really see some of these growths, right? Look at these 2 plates in particular, which I think have lactobacillus on them. These were WLD plates that were blue. Oh, sorry, then moving ahead to day 7, again, we've got more WLD plates that are fully tan at this point. So that's a ton of data, right? And I haven't even shown you all of my HLP tubes. So I have 3 days of these. And let me tell you, I cannot figure out how to properly photograph HLP tubes. So you'll just have to trust me on this. And you can even see on day 3, I tried to take closer up pictures, but there we go, there we have it. All right, so here we are. The actual data in a form that maybe you can understand, I hope. So we have the ID number here. This is the number that I used. If you go back and look at those photos, you'll see that each had a number on them that corresponds to this strain. I tried really hard to get a nice variation of strains. So you can see here we have a lager yeast, we have a regular ale yeast, we have a Kvike, which I think may have had a little bit of Brett in it. We have a mix of 3 Bretts. I bet you don't know what that's called. Let's see, we have this very cool sour producing Saccharomyces, 2 different types of lactobacillus. We've got Brett Brux in here, Pediococcus, and then mix and another Brett. And down here, blanks, of course. So the way you can read this chart, this first section here is microwaved growth. The second section here is pressure cooker growth. And the number you see represents the day in which there was first growth recorded, right? So on day 1, our lager yeast, sac yeast, pretty much everything grew on day 1 on UBA anaerobic. So that's basically best conditions possible for every single brewing yeast, and it grew in every single condition. It did not grow in our blank, so we're really happy about that. All right, there's a few X's here. That's because I chose not to include pressure cooker plates for these particular medias. I was surprised to be given some extra yeast and bacteria from home fermenter centers. So rather than firing up the pressure cooker again, because I hate it so much now that I can microwave, I just sacrificed a few plates and spread them as best as I could apart. Since I already had a few Brett strains up here, I was not worried about if the pressure cooker would pick it up. So sticking on here, now you're gonna have to trust that everything grew where it was supposed to, and nothing grew where it wasn't supposed to. So for example, HLP, which is only supposed to grow lactobacillus and pediococcus, we've got zeros all the way down right here until lactobacillus, pediococcus, and then this lacto mix, right? And that's the same all the way across. A few things I want to point out are where we did see differences in day growth between microwave and pressure cooker, right? So we've got this number 5 here that's highlighted, and this 0, which means it never grew, over here in pressure cooker. Now, it is not rare that Saccharomyces strains are able to grow on UBA in an anaerobic environment. And that's not surprising to anyone who's ever had a beer that maybe had a little yeastiness in it still. Yeast can still survive in an environment without oxygen. They're not very happy, but they can do it. And so we see that the microwave media was able to pick that up, but the pressure cooker was not. Similarly, this Brett Brux down here on this WLD did grow on the microwave, but did not grow from the pressure cooker. And if we look at all of our Bretts, almost all of them grew at least, on day 5. Jump in your head. And then of course, we also have a few instances where the pressure cooker came out on top. This Kvike yeast grew in WLD here, but didn't grow here. It's pretty unusual. That might have been an error just because of how unusual it is. And then this Brett Brux didn't grow here, but it grew everywhere else, even in other pressure cooker medias. So those two are pretty interesting and definitely leaves more to be explored. All right, so what does this mean? Well, microwaving UBA, WLD, and HLP is an effective sterilization technique, and growing common brewing bacteria, it's able to do it just as well as pressure cooking. And then there's maybe a little evidence that can support that microwave media grows some select bacteria at faster rates. We'll see about that. As you can see, there's still a lot more to go. We have not utilized the microwave literally at all in the beer industry. So this is wide open for exploration. We can still look into different pressure cooker and autoclave versus the microwave differences. We could look at their actual biodiversity and say, well, did the sale rate grow faster? Is the density better? Do we have better viability? Are yeast pitches grown on microwaves, the more stable, able to go into further generations. There's a lot still left to be found about using microwaves. We can look at different medias. You know, I chose the 3 that I use the most. I did run some preliminary trials with this diastaticus medium, but I was not really happy with how the experiment ran. So I'm still working on that media in particular. YPD as a yeast growing propagation media, I think that's going to require a lot of exploration moving forward. But then also these other differential medias that are really popular and common in beer labs. And then honestly, you know, looking at other beer spoilers, I have no idea how these would grow. If any of the pitches that I had any sort of infections, nothing showed up. Everything was beautiful, grown exactly the way it should be. So getting pitches of Acetobacter or Enterobacter, Megasphaera zimomios, either any of those, that would be really exciting. And so definitely looking for collabs. 110 plates was really, really overwhelming. So I'm totally down to like share some of the workload if you are. And so just to recap really quick, microwaving media is a smaller footprint, right? A microwave is only a tiny little box that can be put literally anywhere in your brewery, whereas an autoclave either requires a full bench top or stands alone on its own. Same with pressure cooker. Depending on whether you're looking at getting an autoclave or a pressure cooker, it could be 10 to 50 times cheaper to just buy a microwave. It is 2 hours faster. I think even if you take your sweet time in the microwave, I still think you will have, you know, a pressure cooker and an autoclave be at least by a few hours. It's safer for techs and better for your insurance to not have a dangerous potential bomb on premises. And it's safer for your folks to run, or if you're a tech, for you to run. I really hate babysitting my pressure cooker. It's energy saving, right? You can run a microwave on your regular 110 voltage for 8 minutes, or you can run your pressure cooker for a couple of hours, or your autoclave on 210. And it's just as effective at sterilization and growth as these other methods. So if I haven't convinced you yet that the microwave is the brewing lab tool of the future, I think, you know, hit me up in the comments. Thank you all so much for sitting through my talk and listening to me just kind of gush about the microwave, and I really appreciate it. Let me just let you know that February, and I realize that date's wrong now, it's February 18th is the next Fireside Chat, where I will be just kind of talking about the differences between pH and TA. And of course, Dropping Your Acid is my nice little clickbait title. So thank you so much. Please feel free to ask me some questions, and cheers.
Thanks, Dana. Yeah, thank you. I was curious, one of those Kvike strains you said might have had Brett in it. I was curious
if I could tell you which one.
Yeah. Oh, I know which one it was.
Okay. Oh yeah. So what was your, I'm sorry, so then your question.
Could I just like look at that chart that you had with all the the days where you saw the first growth?
There we go.
Yeah. So I'm not a lab guy. UBA, that's Universal Beer Media, right?
Yeah, so that's Universal Beer Agar. And okay. That is it's a catch-all. It really, it will grow pretty much every single thing that grows in beer. And what it basically, its powdered form is basically just like sugar, yeast nutrients, tomato juice, like some of them use beef broth. And then what's unique about UBA compared to all the other medias, powdered medias, is that when you liquefy, when you add water, you also add beer. I use the standard in my lab is Coors Light. And so that's what I use. I like to degas it before use though.
Okay.
Quick question. That was an excellent presentation.
Thank you.
I was wondering if you had any recommendations on types of microwaves or brands or ones that you particularly like.
Yeah, so for this particular experiment, I literally bought it on Amazon. I just got the Amazon Basics $60 microwave. When I first looked into it in December before the holiday rush, it was only $50. So I was a little disappointed that it went up. But I honestly, I think you can do it with anything. All that matters is do your culture flasks that you have fit in the microwave. And that's actually something I forgot to bring up. Do make sure that the culture flasks you are using are a good brand like Kimball or Pyrex, because they do both the cap and the glass needs to be able to be microwaved. Did that answer your question? I think so.
Yes, thank you very much.
Just any old microwave. It's hard not to put the lower range of price for microwave at 0, because I'm fairly certain there's a brewer at every single brewery that maybe has a microwave that isn't in use. I also make it really important to put a sign on your microwave that says not for consumables. If you cook media in there, you do not cook anything else. But I think we'll start to see as this becomes adopted, I think we'll start to see, different standards and stuff pop up. All right.
I'm curious to hear more about the FBI and the pressure cookers. So you're on a list when you buy a pressure cooker new. Is that, I kind of missed that part.
Yeah. So you are put on just a watch list, or I think at this point on Amazon, they keep track of who buys pressure cookers, but it's not actually given to the FBI unless they are asked explicitly. So it has changed. I think it was really, really a big deal right after the Boston Marathon, because they used what is standard in beer labs today. They used All-American Pressure Cooker, which is the brand that I have, because it's made, in the United States with United States steel in Wisconsin. So it's unfortunate that a really excellent tool of cooking and of science was used for something so awful. But that's why. And it is chosen because of its ability to explode, and I think that is one reason to avoid purchasing one to begin with. And I'm very, very excited to maybe sell mine one day.
Do you have any recommendations on tabletop hoods that you particularly like for pouring your media and doing work for aseptic purposes? I
don't. The hood that I have has a long storied story. So it is, and it's a standalone, so it has its own benchtop and is luminaire flow hood. I think there is a company out there that makes some really affordable benchtops that you can set on top of a workstation. If you are limited in space, you don't have a closet or a lab or some place where you can get away in your brewery, one thing you can do is find an area where there isn't a ton of air movement, and you can set up a propane torch or a Bunsen burner to kind of create a little bubble of aseptic area. It is not ideal, so I always recommend having blanks. As you can see, I use blanks in all of my testing to make sure that nothing grows when it shouldn't.
Thank you.
Yeah. All right. Are there any other questions? I think I am going to exit out of the full screen here, or the share screen. And go back to that. All right. Well, thank you all so much for coming and showing up. I know it's a little bit on the late side for these kinds of things, so maybe future iterations will be earlier. But for now, I think we'll stick with the 6 o'clock